My name is Jen Hicks. I'm Deputy Director for the WUSI Human Performance Alliance at Stanford, and also on the team leading the Digital Athlete Moonshot. So in addition to the 2 great sessions we've had so far, the 2 sessions we'll have this afternoon, you'll also see that around us we have a set of about 40 posters and demos that are highlighting research progress and late-breaking results from across the Alliance. And so in this session, this about 20-30 minute session, you'll hear from 10 of the poster and demo presenters who are queued up over on the side. We're ready to go with a set of 2-minute lightning talks. Before they dive in, as Scott showed in his opening remarks, the Alliance was formed by an initial set of 6 partner institutions, but the Alliance network is expanding, and we have researchers from across the country and across the world presenting their human performance research today. And the posters you will see and the talks we've heard so far cover a huge amount of research breadth. When you signed up, we asked you to share 3 words that reflect your passion for human performance research. This slide shows what you all shared. Super fun. So we see things like performance, sport, and health as common themes, which isn't surprising given the shared mission of all of us here today. We also see great resonance around topics like the female athlete, biomechanics, muscle, wearables, and many other vital aspects of performance are also represented. We have molecular and circadian biology, resilience, regeneration, translation. So the posters that you'll see in the lightning talks we'll hear reflect both this commonality and the breadth of the research. This work is really the heart and core of the symposium today and also our Alliance. So with that, let's hear from our lightning talk speakers. I'm going to pass the mic to our first speaker, who's Anthony Gotti. Anthony and each of the lightning talk speakers will introduce themselves, their role in the Alliance, and share some highlights of their poster or demo. Come on up, Anthony. There you go.
Good morning, everybody. My name is Anthony Gotti, and I'm a postdoc here in radiology, and I'm part of the digital athlete team at Stanford. So anterior knee pain is one of the primary reasons in which cyclists seek medical care. Sorry, I didn't go ahead there. There we go. Anterior knee pain is one of the primary reasons in which cyclists seek medical care. And we commonly think anterior knee pain or patellofemoral pain is caused by high pressures between the kneecap or the patella and the femur bone behind it. However, our current biomechanical models don't really model this interaction, and we only look at the patellar or the kneecap's motion as a function of knee flexion. So we don't have variability between different subjects or between how different people are moving. And so in this research, I used a new 12-degree of freedom knee model, shown on the top left of my slide here, where that patella can get some natural motions, and it's really only restricted in mobility by actual ligaments and tendons that are in the knee that didn't exist in the other models. And so I used that model to simulate cartilage pressures inside the knee during cycling. And you can see those cartilage pressures in the top right-hand side of my slide at 4 points in a pedal revolution, where the bright yellow colors are indicating high pressures inside of the knee. And we're seeing those pressures for the patella, the kneecap, as well as the tibia. And so from this preliminary data, we kind of have some findings that we expect. We have high pressures in regions we expect them to be. And so we have starting to get preliminary confidence in this model. In the future, we want to use it to test what are the things we can change to reduce these pressures to potentially reduce injuries. So can we change kinematics or how someone bicycles? Can we change their bike setup? Or really, is this an inherent factor of people's geometry, how their patellar kneecap is shaped or how their femur is shaped? And if you have any interest in biking biomechanics, open sim models, or this new knee model I'm working with, please check out poster number 7. I'm over there, and I'll be at this upcoming session.
Hello, everyone. I'm Shivani. I'm a graduate trainee with the Alliance, and I'm a PhD student in mechanical engineering at Stanford, and I'm advised by Professor Monroe Kennedy. We study how robotic autonomy can be used to restore and improve human performance. So prosthetic arms typically have many degrees of freedom, so many moving joints, and give very little feedback to the user. And this makes it very difficult to perform everyday tasks, even for people who have a lot of experience using these arms. So we've developed a research platform which allows us to investigate the effects of incorporating two features into upper limb prosthesis control. The first one is predicting the next likely action based on cues about the user's attention as well as the nature of the task. And the second is augmented reality feedback to give information about the task to the user while they're performing it. So our platform uses a simulated arm to perform interactions with virtual objects. In this video snippet, the system is calibrated to a user's eyes and it tracks their gaze. So it highlights first an object that they seem to be looking at, and then it plans a motion to go and pick that object up. During lunch, I'll be demonstrating a slice of this work where you can see the arm through the headset picking up the object and completing the pick and place task. In the demonstration, the base is fixed, but in our lab, we use motion capture so that the arm moves along with the person's actual shoulder. So this three-dimensional immersive environment allows us to have a more realistic idea of what goes on when a person controls a prosthesis compared to a simulation on just a screen. But since it is a simulation, it gives us the advantages of being able to iterate our experimental setup quickly and adapt to changing hardware. So we're currently using this platform to compare different methods of prosthesis control with different levels of autonomy. And the goal is to allow people to perform tasks faster, better, more easily, and with shorter periods of training. We plan to make this platform open source. And in the future, we'd also like to make it possible for users to teach actions to their arms. So if you're interested in robotics and augmented reality and their applications in assistance and rehabilitation, and if you'd like to have a demonstration of this arm performing the rest of the task, please stop by Demo 39 during lunch. Thank you.
All right. Good afternoon, I guess. Good morning still, everyone. My name is Nico Philip. I'm a PhD student at the Jayhawk Athletic Performance Lab out of the University of Kansas. And the poster that I'll be presenting today stems from my work with the KU men's basketball team, which just last night secured another Big 12 championship. So a quick rock chalk before we get started. We all know basketball being a sport in which in season the density of practices and games is really, really high. We played last night, Saturday at Texas. Next week, Big 12 championship, so we're playing back-to-back days. It's of importance to practitioners to monitor things such as fatigue, readiness on a regular basis, especially in season. And generally speaking, there's a lack of research that's looking at high-level athletes and over extended periods of time. So within our study, we looked at, from a neuromuscular perspective, we looked at an assessment of neuromuscular performance that is very low risk, easy to implement within the counter movement jump, from which when we perform that on a force platform, we can derive a number of different force time characteristics. Athletes perform these on a near weekly basis over an entire season. We broke the season into a preseason, non-conference, conference, and postseason period and looked at changes and fluctuations over that period. We did see some significant findings, but given the plethora of metrics, if you're interested, I'll be at poster 11. I'll be at poster 11, so we can talk a little bit more about the specific findings, but this could lend insights to practitioners working with athletes to see how neuromuscular performance fluctuates over extended periods of time, such as an in-season period. Thank you.
Hey everyone, my name is Sam Benabou. I'm a second-year undergraduate here at Stanford University studying biomedical computation. Really long name for a major, but basically it's a mix of biology and data science. Last summer, I had the opportunity to be part of the inaugural cohort of the WUTSI Human Performance Alliance undergraduate scholars, where I worked in the neuromuscular biomechanics lab with Professor Scott Delp. There I had the opportunity to design a research project in a field that really interested me, which was seeing how we can use data to learn a little bit more about how people and systems interact. So I'm sure many of us have either used or at least heard of a calorie counting smartwatch. You know, the ones that buzz and flash and throw virtual confetti when you reach your exercise goals. Well, these smartwatches use a combination of heart rate and wrist movement to estimate energy expenditure. But it turns out that how much you move your wrist is not the best indicator of how many calories you expend, as they have errors ranging up to 40%. And being able to accurately estimate how many calories you burn has huge applications in the fields of precision medicine, clinical medicine, and human performance. So my research sought to validate a new wearable technology that doesn't rely on your heart rate or your wrist movement, but instead your leg movement. So with my mentor, Dr. Patrick Slade, we validated this device with subjects performing a range of activities out in the real world, outside the lab setting to simulate a real world environment. We found that our device, our leg kinematics device, had about 40% less error than leading smartwatches in the market. And expanding this device to athletes and high performance teams can really help them target their nutrition and training plan goals. So yeah, please come by my poster number 3 after the talk, and let's talk about it more with all you. Thank you.
Hello, my name is Katie Canals, and I'm a postdoc at UC San Diego. And so I want to tell you about some work I'm doing in multi-scale computational modeling. And this is work that is collaboration with us at UC San Diego, working on the multi-scale athlete moonshot, as well as the agility project at the University of Florida. And so here we're investigating response to isometric contractions in the mouse tibialis anterior muscle. And what we're doing in this project is trying to better understand this connection from exercise that's happening at the whole body level to physiologic responses that's happening in the cells and molecular level. And so in doing this, trying to understand how tissue state, the state particularly of skeletal muscle tissues, is mediating these interactions and what's going on across the scales. And so we're looking at the mouse TA, which is a pretty cool muscle. It has 3 regions with kind of interesting characteristics. And so we're looking at how this very controlled exercise, where we have kind of strict boundary conditions to what's happening in this acute exercise, but how that's going to lead to differences in these regions of the muscle using mechanics-based models, like finite element modeling, to answer questions about how stress and strain might differ in these different regions, and then also looking at molecular differences in these different regions and using systems biology modeling to try to understand some of the molecular mechanisms here. So really starting to connect the dots across scales and kind of develop a more mechanistic understanding of these underlying biological principles that will help us better understand human performance. So I hope you will visit me at my poster over there, number 25. Thank you.
Hello everyone, my name is Damiana Chabarkba and I'm a second-year doctoral student at University of Kansas. I'm also part of Jayhawk Athletic Performance Laboratory under guidance of Dr. Andrew Fry. So, as we probably already heard in some of the presentations before, athletes are exposed to different training intensities, training durations, and recovery periods during their competitive season. Practitioners have been trying to quantify both internal and external training loads during practice sessions and games, therefore. This project is specifically looking at the relationship between internal and external training loads in professional male volleyball players. For the external load assessment, we have used VERT, inertial measurement unit, and for the internal load assessment, we have used rate of perceived exertion scale. We observed that stress metric, external load metric, was strongly correlated with the RPE reported by athletes, as well as the number of jumps that athletes performed during the practice session. However, a weak correlation was observed between active minutes and the RPE score. If you're interested into discussing this in more detail with me, please check out the poster over there, number 17, and I'm more than happy to discuss it with you. Thank you.
Hello, everyone. My name is Megan Keating. I'm the physician associate for the WUSI Female Athlete Program at Boston Children's Hospital. I'm also the chair of the 2023 Female Athlete Conference. My area of research is in female athlete-specific issues, and this specific project came as a result of a clinical question that came from treating female athletes with relative energy deficiency in sport. Menstrual dysfunction is a common consequence of low energy availability in athletes with relative energy deficiency in sport. Resumption of menses is also an important treatment outcome when treating these patients for REDS, but it's difficult to find a specific target when using BMI or percent expected body weight in these female athletes, as many of the oligo or amenorrheic female athletes have a BMI or percent expected body weight in the normal range. So we thought relative fat content might be a useful marker to determine adequate energy storage to support normal reproductive function. And so we did a cross-sectional analysis retrospectively of DEXAs from female athletes aged 15 to 30, basically looking at their body composition and menstrual status. And then we set a percent fat Z-score cutoff of less than -1. And then we compared that to traditional risk cutoffs for BMI and percent expected body weight. While more specific, we found that sensitivity when using traditional cutoffs, such as BMI or percent expected body weight, was poor when compared to using a percent fat Z-score cutoff of less than -1. We also found that sensitivity was better for predicting oligoamenorrhea in this population using percent fat Z-score. And so we found that a DEXA-derived percent fat Z-score of less than -1 might be a good clinical tool to predict menstrual dysfunction in female athletes, and also that a percent fat Z-score of greater than -1 might be a better treatment goal when treating female athletes for relative energy deficiency in sport, but we need to do more prospective work in order to determine the clinical utility. And my poster is 15 if you want to talk more.
Hi everyone, my name is GracePrivett and I'm a graduate trainee working with Dr. Damiana Chabarkba at the University of Oregon. Our lab studies the cellular and molecular mechanisms that underlie skeletal muscle contractile function in humans. Recently, we've been interested in reports that fatiguing exercise, an acute bout of fatiguing exercise, can transiently reduce whole skeletal muscle stiffness. This is particularly intriguing given the fact that fatiguing exercise has also been shown to increase the risk for soft tissue injuries such as to the ACL. So it's reasonable to think that perhaps reductions in muscle stiffness might be contributing to this increased incidence of joint injury. However, the mechanisms resulting in reduced muscle stiffness are unclear. Being a muscle cellular biology lab, we're well positioned to approach this question from the cellular perspective. And we're able to measure muscle stiffness at the cellular level in both active and passive conditions. And this allows us to assess the contributions of distinct intracellular proteins to stiffness. So far, we've found reduced stiffness in both conditions. And this is wild because in this preparation, you're not looking at contributions from altered body temperature or neural input or biomechanical control. Rather, what this suggests is that fatiguing exercise is altering intracellular proteins in a way that results in these reductions. So looking forward, we're excited to continue studying this, as well as to determine whether these cellular observations translate to whole tissue mechanical properties. Additionally, we wonder whether these responses will differ by biological sex or training history. So we're keen on collaborating with fellow members of the Alliance, and we're very thankful for the support of the WUSI Human Performance Alliance. Thank you.
Hi, my name is Mally Gotro. I'm a bioengineering PhD student at the University of Oregon's Knight campus, and today I'll be sharing with you the development of synovial cartilage organoids for preclinical modeling of knee trauma, where following injury, there's an influx of inflammatory cells and cytokines that begin to break down the joint, and over time, this can lead to the development of post-traumatic osteoarthritis, where pain and limited mobility can then decrease human performance, but also quality of life. And as a disease that affects both athletes and younger populations, there's a need for new therapeutics that can prevent or delay the onset of this disease. So where we come in is the development of these in vitro models using cells and biomaterials, focusing on two of the major components of the joint, which are the surface-degraded cartilage and the synovial fluid, or the fluid that lubricates the joint. For the surface-degraded cartilage, we've shown that we can engineer healthy and diseased tissue. And then for the synovial fluid, we're exploring the use of hyaluronic acid to mimic the viscoelastic properties of the synovial fluid, and then exploring its interactions with the inflammatory cells that would be present within the joint. And by systematically applying these components together, we can screen for both potential therapeutics and also explore cell and tissue interactions within the joint space. So for more information or any questions, you can find me at poster 27 during the lunch session. Thank you.
Hey everybody, my name is Aiden Galena, and I'm a PhD student with Dr. Panda Lab at the Salk Institute, as well as at UC San Diego with the Sahu Lab. And I'm here to talk about how large-scale algorithmically ordered data can be used to study exercise at the transcriptome level. So there's a strong but complicated link between exercise, circadian rhythms, and aging. So circadian rhythms are a biological phenomenon that happens periodically throughout the day, and this happens at every level of organization, from sleep-wake cycles at the whole organism level to, sorry about that, to these feedback loops, these molecular feedback loops that happen at the cellular level. This is impactful enough that 80% of protein-coding genes in primates have found some sort of daily rhythmic expression. So exercise is a known stimulus of the circadian regulatory system. And time of day has been found to affect exercise performance, recovery, and metabolic activity. There's evidence that exercise also affects common hallmarks of aging as well. Aging is also known to decrease circadian physiological patterns in humans. And this relationship between exercise and circadian rhythms has been documented physiologically in performance metrics and gene expression. However, a large time series analysis of human transcriptional data is missing to truly corroborate these findings and investigate this relationship at a more aggregate level. So using a newly published algorithm known as chiral to temporally order and sort large-scale transcriptional data sets of human tissue, we explored this interaction between aging, circadian regulation, and exercise in expression. And this interaction varied pretty greatly depending on which tissue we explored. So with this method, we found similarities and differences in metabolic and exercise-related genes and the way they change over the course of a day and a lifetime. And if you'd like to learn more, you can come see me at poster number 9.